too late?
Because I missed the link, sir.
Okay. Then let us start today's talk on probiotics, prebiotics, and postbiotics. Uh, Prebiotics and postbiotics, possibly we discussed very less. Postbiotics, we didn't talk much. So let us start from the postbiotics. What is it? Postbiotics, we know the probiotics, living microorganisms that inhabit the host, eukaryotic host, and that exert beneficial effects to the host in different ways. They are called probiotics, and prebiotics are non-living uh, materials, especially polymeric substances that enhance the growth uh, of and colonization of the probiotics. And we also know symbiotics. Symbiotics means probiotics and prebiotics. Now let us discuss a bit about, I didn't discuss possibly in this class, postbiotics. What are postbiotics? Postbiotics include any substance released by or produced through the metabolic activity of the microorganism which exerts a beneficial effect on the host, directly or indirectly. As postbiotics do not contain live microorganisms, the risk associated with their intake are minimized. That means postbiotics, they are not living organisms. They are the substances produced by the living organism through the metabolic activity. Substances can be small molecule, can be big molecule, but those substances should exert beneficial effect on the host. And if you go this link, you can find a fantastic review article on uh, postbiotics. So please go through, and I also shared this link on your uh, Facebook group. So probiotics, uh, prebiotics, probiotics, and postbiotics. You see, in total microbiome in the eukaryotic host is depend on the interaction or effect of these three pillars. And exercise, healthy diet obviously is necessary. So if you have good probiotics, good prebiotics in your diet, and if you take postbiotics, probiotics also produce postbiotics, but additionally sometimes postbiotics also, offered to and can be taken because commercially they are available. And if you have good exercise and healthy diet, then your microbiome would be balanced. And if your microbiome is balanced in your body or in the body of any eukaryotic organism, uh, the eukaryotic organism would be healthy and immune. So here you can see the center, you can see the probiotic bacteria, and the probiotic bacteria, what kind of non-living materials they can produce. For example, after their death, cellular wall fragments, these are non-living substances, so they can exert some beneficial effects to the host. So they are, postbiotics. And exo polysaccharide. These bacteria, excrete different polymeric substances like exopolysaccharide for making their biofilm or like that. So these exopolysaccharides are also postbiotics. Bacteria obviously use diverse enzyme to degrade the organic and inorganic substances to get their energy, as well as to compete with other organisms. So enzymes are non-living and they are postbiotics. different vitamins, phenols, and different compounds, short chain fatty acid they can produce. And they are also good for the health of uh, host. So they are postbiotics. So bacteria lysate, if you kill the bacteria, and then a broken part of the bacteria, if you want to use, they can be used as postbiotics. Postbiotics sometimes can be used as a vaccine in some cases, because non-living bacteria, heat killed or other chemically killed bacteria sometimes used as vaccine. And supernatant, when you culture bacteria, you can find bacterial cells as well as the culture fluid. So if you do centrifuge, you can separate the bacteria, and supernatant contains lots of metabolites and enzymes. So these are the methods of acquisition of postbiotics. And the concept of postbiotics conceals either metabolites or fragments of microorganisms which confer a beneficial effect to the host. The structural heterogeneity, of bacterial cells, of postbiotics implies the abundance of possible techniques used to probiotics acquisition. Lysis of bacterial cells may be achieved by chemical or mechanical techniques. These methods include enzymatic reaction, solvent extraction, sonication, and heat extraction. Dialysis, chromatography are used to isolate and identify desired molecules. For example, from the supernatant, if you want to purify the desired molecules, for example, a small secondary metabolite or an enzyme, you need to do different kinds of chromatography, and then you need to do a apply spectroscopic techniques to elucidate the structure of the molecules. Short chain fatty acid, vitamins, and phenols, they are also can be purified by using chromatographic technique, and structure can be elucidated by the spectroscopic method. Chromatographic method include, a thin layer chromatography, silica gel chromatography, or many other chromatographic technique. And spectroscopic method include mass spectrometry, NMR, nuclear magnetic resonance, circular dichroism, CD, and others. So the question is how the postbiotics act on the host. Postbiotics, they can modulate the immune response of the host. For example, I told you some, lysate of the cell or killed bacteria, pathogen, killed pathogen. If you can use uh, to the host, then host can sense by their surface, cell wall and others. Host can sense the uh, presence of the pathogen. In that case, host immune system activated, and antibody and other immune responses are taken place. So in you can use postbiotics just like a vaccine. Infection prevention, similarly, infection prevention, if immune response is activated, then infection obviously being prevented. Lipid, cholesterol metabolism, some of the postbiotics, they are very helpful for lipid and cholesterol metabolism. As a result, homeostasis in the lipid profile can be maintained. Anti-tumor or antioxidative activities, some of the postbiotics, they are anti-tumor compound, or some of them are like phenolic compounds, are antioxidant activity. They can neutralize the superoxide produced in the metabolic process. So they are very, very helpful, and some of the postbiotics can be used just like a medicine. So, mechanism is now almost clear. They exert their mechanism, that means beneficial effects to the host by immunomodulation, infection prevention, lipid cholesterol metabolism, anti-tumor and oxidant activities, and even antimicrobial activities. That means other pathogen can be suppressed. So these are the good effect of postbiotics. Postbiotics are now, commercialized in many ways, although not available in our country. Prebiotics, possibly I discussed about the prebiotics, their classification, not digested, that or only partially digested, not absorbed in the small intestine, poorly fermented by bacteria in the oral cavity, well fermented by seemingly beneficial intestinal bacteria, poorly fermented by potential pathogen in the bowel. That means there are different kinds of prebiotics, and they are all non-living materials. And I am not sure whether I discussed this once in biotics.
Yes, sir, you have
Already,
discussed this.
already I discussed, yes. The mechanism of beneficial effects of probiotics on immunological function remains unclear, not fully understood. Several possible models have been proposed. Prebiotics are able to regulate the action of hepatic lipogenic enzymes by influencing the increased production of short chain fatty acids, such as propionic acid. The production of short chain fatty acids, especially of butyric acid, as a result of fermentation, was identified as a modulator of histone acetylation, thus increasing the availability of numerous genes for transcription factors. The modulation of mucin production. It was also demonstrated that some of the prebiotics and several other prebiotics cause fructose oligosaccharide and several other prebiotics cause an increased count of lymphocyte and or leukocytes in gut associated lymphoid tissue and in peripheral blood. The increased secretion of IgA and gut associated lymphoid tissues may stimulate the phagocytic function of the intra inflammatory macrophages. That means, in case of animal model, these five possible mechanisms are being known by the action of prebiotics, but precise information for all the prebiotics has huge diversity, still been not fully understood. This is why research is necessary and many research are going on. But anyway, prebiotics can exert beneficial effects to the host by various ways, and it depends on the chemical composition of the prebiotics as well as the host where you are applying. And prebiotics, in case of other than animal model, prebiotics also enhance the colonization of the microorganisms, because as they are fermented by the probiotic organisms. So in case of plant, there are several literatures that probiotic, when we apply to the plant, in most cases they fail to compete with the uh, microorganisms available in the environment. In that case, if you add the prebiotics like chitosan or like that, other polymeric substances, then you can, have the uh, desired activity, because of the prebiotics enhance the colonization, prebiotic enhance the uh, virulence of the probiotics for colonization as well as fighting against other microorganisms to sustain in the applied, plant organs like rhizosphere or others. So benefits of symbiotics. Symbiotics have the following beneficial effects on human. Increased lactobacillus and bifidobacterium genus count and maintenance of balance of the intestinal microbiota. Bifidobacteria and lactobacillus are the major naturally occurring probiotic beneficial microorganisms in the human intestine. Improve hepatic function in patients suffering from the cirrhosis, improved immunomodulative abilities, and prevention of bacterial translocation, and reduced incidence of nosocomial infection in patients post-surgical procedures and similar interventions. These are a few, examples of their beneficial effects. But if you go to the literature, you can find lots of new informations are available. So these are all about the basic concepts of probiotics, prebiotics, and postbiotics, and also symbiotics, and their different classification, beneficial effects to the plants and environment. Now we can have, larger discussion. Uh, please ask some question.
Sir, prebiotics, probiotics, and postbiotics both have good function on the health. In terms of, if I say postbiotic, just are the metabolites, and uh, I think they do not show the antimicrobial substance. But in case of prebiotic, in case of probiotics, they uh, secrete antimicrobial substance in the human gut. So will they? Which one will be better uh, in terms of health benefit, probiotics or postbiotics?
Yeah, it's a good question. In case of probiotics, they are living organism, and they are always active in the niche. That means the place where you applied, and they have the ability to colonize. They have to ability to fight for the nutrition competition for the nutrition. They have ability to suppress other microorganisms. By producing different lytic enzymes and antibiotics or others mechanism, and in the presence of colonization of the on the host, they also trigger the gene expression, because living cell interaction is different than non-living materials and living cells. So probiotics, if it is colonized in optimal level, and if it is maintained, then it is obviously a long-term benefit. But in case of postbiotics, they are non-living materials, obviously a product of the probiotics. In that case, the amount you applied, this amount will be depleted in course of time. Because they cannot proliferate themselves, they cannot, reproduce themselves. So postbiotics can contain complex mixture of different, biochemical substances, polymeric substances, even phenolic substances, fatty acids, or even antibiotics. But whatever uh, uh, present in the postbiotics, you have to understand that you have to apply postbiotics after certain interval, like you are taking the antibiotic. When you take an antibiotic or any drug, then drug molecules work on the target organism, and gradually, metabolized, and after certain period the action eliminated. This is why in case of antibiotic you need to take a certain course and to maintain a certain dose. Postbiotics also similar, so you need to maintain a certain dose and a certain course because they are non-living. So there are lots of differences. So I shall give you a small assignment for next class. Compare and contrast probiotics, prebiotics, and symbiotics. Compare and contrast. So there are some differences. There are some similarities also. So please make a table. Compare and contrast probiotics, prebiotics, symbiotics, and postbiotics, so that we can get all the information. For example, structurally, probiotics are living, uh, 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 prebiotics are non-living, symbiotics are living, non-living, living plus non-living, and postbiotics are non-living. So the functional aspects, they have also some differences because prebiotics and postbiotics, they cannot colonize. Actually, they are basically metabolized by the different organisms living in the place or ecosystem where you are applying, for example, intestine or the rhizosphere or any other places. And they can be used as the growth stimulator or even nutrient source of the probiotics. Sometimes they are fermented. So there are lots of differences, and there are lots of similarities also, because none of them are actually synthetic. Most of them are naturally originated materials. Some are living and some are not living. Any question?
I want to ask one question. How can we understand when we need prebiotics or postbiotics or symbiotics? Because there is any symptoms.
Yes, in case of human, sometimes the people who are suffering from obesity, doctors suggest probiotics, prebiotics, or symbiotics like that to tackle the obesity, because obese people have different microbiome in their guts. So this is one of the symptoms. Even after taking antibiotics in many countries, a long course of antibiotics can even kill the uh, probiotic microorganism present in the guts and in the body. So human suffer from the defense mechanism supported by the probiotics. In that case, after taking the antibiotic, doctors sometimes suggest probiotics, prebiotics, symbiotics, or something like that, so that the microbiome, beneficial microbiome in the guards can be restored, or their population can be maintained, because they are the continuous guarding us against the different organisms. And in case of plant science, when you apply probiotic microorganism or fisheries or other disciplines, uh, probiotics, uh, uh, usually we use as an alternative of synthetic materials, for example, fertilizer or pesticide. In that case, probiotics and prebiotics together being used, so that the probiotics you are using, they get a better environment and better nutrition to fight against the other environmental microorganisms, so that they can colonize in the niche at a better rate, and can dominate in the target site, for example, rhizosphere, phyllosphere, or others. So these are the strategy, but clear-cut symptom, in case of if someone has probiotic, very poor probiotic microorganisms, in that case, harmful bacteria, for example, Helicobacter pylori can, capture the intestinal layer and colonize there, and Helicobacter pylori can cause cancer. They can produce toxin. So people suffer from uh, uh, different gastrointestinal problem like gastric or other problem. So there is a test. You can test whether, even in Bangladesh, you can test if you go to ICDDRB, you can test whether you are infected by the Helicobacter pylori or not. And there is a kit, that means a cocktail of antibiotics for a few days if you take, and you can clean your intestine from the Helicobacter pylori. This is one way, and if you clean your intestine after by the using of kit, that is antibiotic, then you can take, in many countries doctor suggests to take lactobacillus and bifidobacterium and some other prebiotics, so that your microflora in the intestine can be restored with beneficial ones. Any more question? Any idea? In Bangladesh, poultry industry use probiotics, even fisheries, especially the shrimp industry, they use lots of probiotics, but those probiotics really active or not, we don't know, because they are coming from other countries, and probiotic microorganisms are sensitive to the environment. In that case, if you import from other countries, there is a high risk of their inefficacy in the new environment. So best idea is to discover probiotic bacteria from the shrimp we are cultivating in our ecosystem, or the poultry we are cultivating in our system from the poultry gut, and then you commercialize them and apply in our country, then surely they will exert the beneficial effects. But the probiotics, especially in shrimp industry, we are using just by force of the European Union. Otherwise, they will not accept our exported shrimp if we use the antibiotic. These are the very fascinating area to do the research, because probiotic has growth promoting effect already been established, and they are powerful alternative to the antibiotic. Any more question?
No, sir.
No, sir.
In our next class, we shall discuss in detail about probiotic microorganisms that we are exploring in our laboratory, and we have some good work. And Ruhul, you can have new information regarding the plant probiotics. So we shall discuss in our next class plant probiotics, because plants are also eukaryotic organisms. So fundamentally, the function of probiotics in different organisms are same, but in case of plants, there are some differences, because plants, the green plants, they are the only organisms in this planet that can trap the solar energy and convert them into chemical energy. And all the living organisms in this earth, including human, they are directly or indirectly depend on the chemical energy synthesized from the solar energy by the photosynthetic process. So plant are unique organisms and very powerful organism. And if there is no plant in this earth, there is no chance of having other, eukaryotic organisms in this earth. Some bacteria and some microorganisms which can get energy from the environmental uh, system, uh, uh, they may survive, but all eukaryotic organisms cannot survive in this earth. So I hope you will enjoy that talk also. Any more question, Shafi?
No, sir.
Shafi, did you communicate with Dipali for the uh, gene expression and regulation course?
No, sir. But I got some message from
Okay, please contact with her because today's at 2 p.m. The course is supposed to be delivered by Dipali Rani Gupta. So please, both of you, two students are there, Nila and you. So communicate with Dipali. I informed her, and please keep continue with her because she is expected to leave Bangladesh for doing her postdoc in Japan in early March. And if she leave, then after her leaving, I shall continue the course. So it is better to have her lectures first. Thank you so much.